আমাদের শরীরে একটা রেগুলেশন হচ্ছে বা কোনো একটা চেঞ্জ হচ্ছে, সেটাকে আমরা রেগুলেট করছি। একটা এক্সাম্পল দাও, যে কোনো। Give one example where any metabolic function is being regulated in our body. Any metabolic function. বল, তুই বল। Which we can answer through gene regulation. So gene regulation is not an alien thing, it is happening in our body. So give one physical example. তাহলে কাউকে চড় মারলে কাটছে, কাউকে চড় মারলে রেগে যাচ্ছে। So who is secreting adrenaline? And who is telling you to secrete more adrenaline, secrete less adrenaline, secrete more endorphin, secrete less endorphin? Right? These all are regulated. See, when you see biryani, you start salivating. Is it a gene regulation? Gene regulates secretion of hormones. Hormones regulate metabolic function. How? By increasing or decreasing synthesis of certain enzymes. This is the pathway, simple pathway. খাবার দেখলে যে স্যালাইভেট করে, সেটা হচ্ছে neural reflex. So regulation, gene regulation is different from neural reflex. যেমন আমি যদি এখন এটা ছুঁড়ে মারি, কি হবে? Is it gene reflection? No. It is neural reflex. The person who can master this neural reflex, he can be Virat Kohli or Sachin Tendulkar. When Shoaib Akhtar sends a ball of 150 kilometers per hour speed, then usually we will do like this, except Sachin Tendulkar, because he has mastered his nerves. He has control over his nerve. He can control his reflexes, neural reflexes. So gene regulation is different, where secretion of hormone is regulated, and secretion of hormone regulates the synthesis of certain enzymes which bring about metabolic changes. Now, this is the usual central dogma of molecular biology. All these black lines are the regions where a gene can be regulated. A gene usually reside on DNA. Why I said usually? Because there are many RNA viruses who has no DNA. That's why I said usually gene resides on DNA. The DNA is synthesized on itself, called replication. Right? Then if replication process is inhibited, then gene multiplication will be inhibited. You have more number of DNA copies, you have more number of copies of genes, you have more production of the same gene product. So if you inhibit replication, you strike down the production of certain gene. So I said gene regulation can be performed on each and every black line. Correct? So this is how we can regulate replication. We can regulate transcription. Can we regulate reverse transcription? No. All the medications against RNA viruses are reverse transcriptase inhibitors. Translation from RNA to protein we can regulate. Correct? So all these 1, 2, 3, 4 regions we can regulate. Now, organism to organism, this may vary. See? What is the difference between these two structure? Key difference, bolo? A হচ্ছে prokaryotic, eukaryotic. কি করে বুঝলে? How did you understand? Nuclear membrane is there. A distinct nuclear membrane is present. Distinct nuclear membrane. If it is not distinct, then what do you call this? Nucleoid. Right? So distinct nuclear membrane separates prokaryote from eukaryote. Now, if you see the replication, transcription and translation in prokaryote and eukaryote, then a single liner difference is this. What is that? That transcription and translation in eukaryote, transcription and translation in eukaryote is separated by space and time. Sounds a little bit difficult. Space and time. It is different by space and time. Means what? See, here transcription and translation occur simultaneously. Where? In prokaryotes. Why? Because there is no nucleus. There is no nuclear membrane. Whereas in eukaryotes, the mRNA or hnRNA or primary transcript, whatever is produced, should undergo post-transcriptional modification so that they can produce mRNA, tRNA, rRNA, snRNA, and so on. Now, these RNAs have different functions, and these modification of RNAs takes place within nucleus, that too a region called nucleolus. So, in case of eukaryotes, first the mRNA is modified, then it is secreted from nucleus to cytosol. So, these two steps are involved. So, transcription occurs simultaneously with translation in prokaryote, whereas for eukaryote, translation to occur, it requires first modification of mRNA followed by transfer of mRNA from nucleus to cytosol. That is why they are different in time and space. They are not taking place at the same time. Number one. Number two, they are not taking place in the same space. Transcription occurring in nucleus, translation occurring in cytosol. That is why they are differentiated by time and space. অনেক বড় জিনিস ছোট্ট করে ওয়ান লাইনার হচ্ছে এইটা. Now, how these genes are regulated? The first thing we come to know is Operon. Jacob and Monod, these two people, they first came up with the concept of Operon. Operon means a cluster of genes which operates simultaneously. They reside together and they are regulated simultaneously. Whereas in eukaryotic gene regulation, it is either of two types, either short term or long term. Short term regulation, example, we eat food, blood glucose in the serum, blood glucose level in the serum increases, plasma increases, and insulin secretion increases. This is short-term regulation. Long-term regulation, when insulin is not being secreted by the by the pancreas, like in case of diabetes mellitus, then other things happen. Correct? Usually the renal threshold for normal person is 180 milligram percent. But a person who is suffering from prolonged period from diabetes mellitus, he shows an elevated level of renal threshold. That is due to what? A long-term change under diabetic condition. Okay? So long-term gene regulation. One more example you can give. The people who are residing in the center of the earth, central region of the earth, they receive more sunlight, they become darker. Those who reside in the higher altitude, they become more fairer because they receive less amount of sunlight. So over the years, the body has changed to produce less melanin so that they can receive more sunlight. Now, concept of operon. So prokaryotic regulation can be done by using Operon model. Operon can be regulated by two ways. Number one, repression and derepression mechanism. Number two, attenuation. Both the ways are to regulate Operon models. One is using repression and derepression mechanism. Another is using attenuation mechanism. What is repression and derepression mechanism? In first mechanism, repression and derepression mechanism, the suitable example we use lactose operon or Lac Operon, and in case of attenuation we use TRP operon or Tryptophan Operon as example. The lactose operon or Lac Operon example debo in case of repression and derepression mechanism model. Attenuation will give TRP or Tryptophan Operon. Now Lac Operon, first we have to know about the Lac Operon. What is the structure of Lac Operon? What are the structural genes present in the Lac Operon? Like a general structure of operon must include these three parts: regulatory gene, promoter and operator region, and structural genes. The structural genes are a series of genes located in a cis configuration, means in the same DNA, cluster of genes. Number of more than one genes are located in this together. Promoter and operator just precedes, just ekটু ওর আগে থাকে, precedes the structural gene. The promoter and operator means the promoter region is a region which is recognized by transcription initiation factor. What is that? Sigma factor. Correct? If it is prokaryote, then it is sigma factor. And operator region is the region where it binds. Correct? So, in some genes, promoter and operator may be separate. In some genes, they might be overlapping. In some genes, they might be overlapping. Correct? Then, before the promoter operator region, there lies a regulatory gene. This regulatory gene is actually a gene which produces repressor molecule. Means, what is repressor molecule? Repressor molecule is a trans-acting factor. Repressor molecule is a trans-acting factor which inhibits production of operon genes by binding at operator region and thereby physically hindering the binding of regulatory gene is a gene which produces repressor molecule. Stop. Repressor molecule is a trans-acting factor which binds at the promoter-operator region and thereby inhibit the production of structural genes products. It does so by physically hindering the binding of transcription initiation factors or sigma factors. তাহলে repressor molecule কাকে বলবো? এটা একটা trans acting factor. Promoter operator region, এগুলি কি? These are cis acting elements. Remember these two words, cis acting elements and trans acting factors. Elements are sequences of DNA. Elements are sequences of DNA. Factors are proteins. So, repressor molecule is a trans-acting factor. Promoter operator region is a cis-acting element. Cis-acting elements, trans-acting factors. Cis-acting elements, trans-acting factors. So, if I say it's a cis-acting element, it binds with transcription factors so that TF2D factor can bind transcription initiation site more tightly and more precisely. It is the presence of GC box which determines that whether the gene needs to be produced more or less. Anyway, so this regulatory gene, like every gene must contain the structural part, the operator part and the promoter part. So this is regulatory gene I, and it has got a promoter also, PI. Right? So regulatory gene has got one promoter, then followed by gene, then followed by promoter operator, then followed by structural gene. This is a general structure of operon. Now we will discuss the structure of Lac Operon. This structure of Lac Operon. The Lac Operon from left to right, see PI, then Lac I, then P/O, promoter operator region, then Z, Y, A, Lac Z, ore babare, Jolga. When it is about the gene, the portion on DNA, it is written in small letter. When the product is formed, then it is called the product of Lac Z. The product of Lac Y. The product of Lac A. The product of Lac I. Correct? So the product is written in capital, starting with capital, whereas the genes present in the DNA should always be written in small letter. By writing so, we can express the meaning. We need not to say it is gene, it is a product. Right? So what does it produce? Lac Z produces beta galactosidase enzyme. What does it do? This enzyme is responsible for breakdown of lactose. Correct? Y codes for permease enzyme, which allows the entry of lactose into cell. A has got acetylase, trans-acetylase activity, which is irrelevant to this Picard, which is irrelevant to this lactose metabolism. Now, why lactose? Glucose is the most preferred carbohydrate, but when glucose is absent in a medium, then the bacteria must rely on other sources. What is other source? We keep the milk open in open air for some time. After a few hours, it gets spoiled. Why? Because bacteria feed on lactose. So the milk contains lactose, not glucose. But bacteria's internal system can metabolize only glucose, not lactose. Our cell membrane has got transporter for glucose, GLUTs, GLUT 1, 2, 3, 4, but no transporter for lactose. But here the bacteria, if it has to survive, it must change its food from glucose to lactose. So the bacteria is now changed to glucose to lactose. But how the lactose will enter? For that they need a new transporter, permease, which will allow the lactose to enter. But the after entry, it must be broken down into glucose and galactose. If we break down glucose and galactose, then we can metabolize it easily, the bacteria. Otherwise, we have to keep a separate set of enzymes, whole separate set of enzymes for lactose metabolism. Instead, what we do, the bacteria, it just breaks the lactose to glucose and galactose and then utilize with the same pre-existing enzymes. Correct? So, to utilize lactose, it requires lactose. Correct? To utilize lactose, it requires lactose. Why? Because when there is no need of lactose utilization, a lot of glucose is present, then this gene is inhibited. It does not produce any of these products. Trans-acetylase has no relation with the lactose metabolism so far. Okay. Now, these two products are required for lactose entry and metabolism, but it is not required when there is lot of glucose present in the system. So, that time it should be stopped. How? By producing this product, which will go and bind here and which will disallow, physically dislodge the initiation factor, the sigma factor. The sigma factor, it cannot work, it cannot bind to the promoter operator region. So there will be no transcription. Now, the same model, but the same example with a different model. This is, now, modified, আচ্ছা এইটা একটু পরে আসছি, আগে এটা দেখি। Let's check this out. This is important. This is important to understand. See what are the conditions and what is the effect. If both glucose and lactose are present, then do we need to produce Lac Operon products if glucose is present? No. So no transcription. If both glucose and lactose are present. Now glucose is present, lactose is absent. Again, after all, glucose is preferred. So as long as glucose is there, no transcription. Glucose and lactose both are absent. Neither glucose nor lactose. So there is no point in making enzymes which can utilize lactose. If we do not have the lactose in the medium, why should we produce the enzyme which will be required for lactose metabolism? No required, no transcription. But when lactose is present but glucose is absent, then it performs the transcription so that lactose in the medium can enter into the cell and then it can be metabolized to produce energy. Correct? Now, how this happens, this is explained in this diagram. This diagram shows everything and it is a self-explanatory diagram. You need to remember this diagram in Lac Operon and you have to explain this by drawing this diagram. This diagram shows how a repressor molecule actually regulates the gene expression of Lac Operon. Correct? The first part shows how repressor molecule blocks the entry or binding of transcription initiation factor so that no Lac Operon genes are transcribed. The second part shows how repressor molecule is dislodged by one activator molecule. What is that? The repression molecule usually is a tetrameric compound. It is active when it is tetrameric form. It is inactive when it is in dimeric form. The tetrameric form can bind to a specific region of operator. The operator region has got three distinct regions: operator 1, operator 2, operator 3. So within these three regions, the repressor molecule can bind operator 2 and 3 and does not allow the sigma factor to bind. Good. When it is required to be synthesized, the Lac Operon genes, then the first thing is to be done is to dislodge repressor molecule. How it will be done? By producing certain activator molecule. What are those? The naturally acting activator molecule is allolactose. Allolactose. The artificial one is IPTG, isopropyl thiogalactoside. If you are willing, you write. If you are not willing, you don't write. It is your right to write or don't write. Huh? Use your right for not writing or writing. I am no one to write out your right. বলো প্রশ্ন আছে কিছু কোনটা আইসোপ্রোপাইল থায়োগ্যালাক্টোসাইড IPTG এটা হচ্ছে আর্টিফিশিয়াল অ্যাক্টিভেটর এটা কি করে they bind with repressor molecule and thereby they don't allow the repressor molecule to bind operator region. So in absence of repressor molecule in the operator region, the transcription initiation factor, the sigma factor, can bind to the operator region and can initiate the transcription of structural genes of Lac Operon. Right? Fine. So initially, production of repressor molecule is very important. Now, the question may arise that what came first, the egg or the chicken? The egg or the chicken, correct? So, if lactose was absent, then how come the first lactose molecule entered? Because no permease will be synthesized. Question or not? And if lactose was present, then why there is a need to produce permease again? How come it will enter arolactose. Without arolactose, it cannot produce other enzymes. And arolactose is produced from lactose. So who brought the first lactose? Who let the dogs out? So who let the first lactose in? That is the question. The answer to this question is very easy. Whenever we cannot explain our mistake, we say what? Suppose you made a beautiful wall, and then after some days, you see the water is being seeping through the wall. What is the answer? Leak. And suppose you are talking with me, and suddenly you utter some abusive word, which is very commonly used. And then you say, sorry sir, my slip of tongue. That means what? Leak. So when we do not have any answer, we say leak. So something like due to some leakage in the cell membrane, small amount of lactose might have entered in the time way back, and the cell has remembered, yes, that was lactose, and produced some arolactose. It was there to produce more and more Lac Operon genes. Correct? So this is still unanswered. If you find the answer, Nobel Prize is waiting for you. Fine. The similar repression and derepression in case of tryptophan operon, I must explain. The tryptophan operon shows the regulatory gene like LAC-I, it is PI, then followed by TRPI. This is an inactive repressor molecule which must bind to the operator region, and then it should block the further transcription of this gene. Now, unlike Lac Operon, tryptophan operon has got 5 genes. These 5 genes lead to production of 3 enzymes. These 5 genes lead to production of 3 enzymes. 5 to 3. 3 enzymes, the enzymes which are important for biosynthesis of tryptophan. We, unlike us, the bacteria can produce their own tryptophan. So the biosynthesis of tryptophan is dependent on production of these 3 enzymes, which is dependent on production of these 5 gene products. So naturally we can say that if tryptophan is abundant in the medium, then these synthesis will be stopped. If tryptophan is absent in the medium, then the synthesis will again resume, and tryptophan can be synthesized inside the cell and they can survive. Fine. So that is regulated by this repressor molecule. If tryptophan is present in the medium, then it binds with the repressor molecule and it becomes activated repressor, and then it binds and blocks the further transcription. That means if medium has got excess of tryptophan, then it goes, binds with the repressor gets activated, and it binds with the arolactose. Does the opposite way. Lactose becomes arolactose binds the repressor becomes inactive, and when there is no lactose, arolactose is produced. When there is no lactose, arolactose is produced. Arolactose binds the repressor cannot bind the operator. Here opposite. Tryptophan is abundant, no need to produce tryptophan operon genes. So tryptophan binds the receptor gets activated, it binds the operator region and blocks the. Right? So we will stop here because the next part is a little bit difficult, complicated. So usually we teach in the same 1 hour class, but anyway we have a lot of time, so tomorrow also You can continue. Tomorrow your class is there or already declared?